Welcome. It is my pleasure to welcome you today to Susan G. Komen's virtual MBC Impact Series event. My name is Kelsey Hampton. I'm Komen's Director of Mission Communications and Education, and I'm here on behalf of Casey Volpe, Komen's Manager of MBC Initiatives at Komen. This programming for the series is envisioned as a year-long conversation with attendees, presenting new information and speakers every month. Today, our topic is metastatic lobular breast cancer, and our featured speaker is Dr. Steffi Oosterrijk. Before we begin, Komen would like to thank our sponsors for supporting the 2024 MBC Impact Series. Thank you to our premier sponsors, AstraZeneca, Gilead, GE Healthcare, Merck, and our signature sponsors, Walgreens and Daiichi Sankyo. This series is made possible because of their generosity. One more housekeeping note. While our speakers are talking, please feel free to post your questions in the Q&A feature in Zoom, and we will answer as many of these as we can. Please remember that our speakers are not able to provide individual medical advice and are only speaking to their experiences. Towards the end of our programming, a survey link will be placed in the chat. Your feedback is very important to us and is so helpful as we plan future MBC programs. Now, we are excited that you all are here to join us for today's discussion. And to start, I would like to introduce you to our fantastic moderator, Mary Domenecki. Mary, off to you.
Hi, everyone. I'm delighted to be here. As she mentioned, my name is Mary Domenecki. I'm actually an 8-year survivor of HR-positive, HER2-negative stage B lobular breast cancer. I'm an advocate with Komen Advocates in Science and the Lobular Breast Cancer Alliance. I'm also an Imerman Angel and have reviewed grants for the Department of Defense CDMRP program. In my day job, I work in healthcare market research with clients developing oncology products. I am a science PhD by training. I worked in microbiology as a graduate student at NYU School of Medicine and designed and tested cancer vaccines in my postdoc at University of Pennsylvania. I'm also mom to 3 boys, ages 19 to 25, and married to my college sweetheart for 29 years. I'm delighted to introduce our speaker today, which is Dr. Steffi Oosterrijk. Dr. Steffi Oosterrijk is a professor of pharmacology and chemical biology, co-leader of the cancer biology program at UPMC Hillman Cancer Center, HCC, and co-director of the Women's Cancer Research Center at McGee Women's Research Institute and HCC. Since her early graduate studies in Germany, Dr. Oosterrijk has been passionate about breast cancer research. She enjoys working in multidisciplinary teams, hand in hand with medical oncologists, surgeons, bioinformaticians, and pathologists, motivated by important clinical problems with critical input from breast cancer advocates. Dr. Oosterrijk's Komen-funded research is focused on understanding the spread of ER-positive breast cancer by analyzing liquid biopsies and metastases from a tissue donation program in order to predict drug resistance and progression, with a special focus on invasive lobular carcinoma and tumors with estrogen receptor mutations, as well as working with a group of patient advocates to implement rapid autopsy programs at other institutions for tissue donations. Please join me in welcoming Dr. Oosterrijk.
Thank you, Mary, for this kind introduction. Thank you, Casey. Thank you, everybody from Komen, to give me a chance here to present today updates on metastatic lobular breast cancer. And let me share my screen. So yeah, it's a pleasure to present here today on metastatic lobular breast cancer. This is my conflict of interest, and none of them are of relevance for this presentation. So I thought I would give you a brief overview of what I'm talking about today, about, brief introduction about myself, how I started studying lobular breast cancer, an overview in general about ILC biology. Then I will talk about unique aspects of lobular metastasis, some words on liquid biopsy, on a rapid autopsy program, and also on modeling ILC. And I will end with conclusions on outlook. Brief introduction. Mary already said that, I got my PhD at the Humboldt University in Berlin, did my postdoc in San Antonio, Texas, was an assistant associate at Baylor College of Medicine, and since 2010, I've been at Pittsburgh here. I'm professor, co-director of Women's Cancer Research Center, and co-lead the cancer biology program at Hillman. I started to work here on lobular breast cancer, and that was really somewhat coincident that we identified a pathway which turned out played a role in lobular breast cancer. And the pathologist here, David Dabbs, was so excited to really share his expertise and curiosity in ILC. And then we realized there wasn't much published. We were very, very surprised. So, this is how a lot of it started. After studying this for 6 or 7 years here in Pittsburgh, we decided to get researchers and clinicians who had an interest in lobular breast cancer together, where we organized the first international ILC symposium, which is now 8 years ago. And at the time, we also invited patients and patient advocates. And make a long story short, I think a lot came out of this original meeting, not only on the scientific side with respect to collaborations, but also for starting advocacy. I think the patient, and this was actually a conclusion at the meeting from the patient, that they needed quality information about lobular breast cancer to inform self-care, that there was a lack of confidence in therapies which were really originally tried and tested in ductal cancer, and they were very eager to bridge gaps, support research, and raise awareness. I've been very, very fortunate over the years since that meeting where I met Lee Pate to work with her until her unfortunate death now almost 2 years ago. She was a really strong, fierce ILC patient advocate. She was the founder of the Lobular Breast Cancer Alliance and really loved to work with Komen. She was a Susan G. Komen a steering committee member and a Komen scholar. One of the things I worked with her very hard on is to really put on paper how researchers, clinicians, and patient advocates can work together to accelerate lobular breast cancer research. And now, a decade later, there are a number of organizations where patient advocates, not in the US, but also in Europe, have come together and really drive forward research in lobular breast cancer. So what about, in general, ILC biology? This is a quote from a review which was written up 50 years ago, and these were absolute leaders in the field. A review on lobular carcinoma of the breast. I don't want to go through it in detail, just to say that quotes from there are survival rates, as are other aspects of the natural history of this tumor, are the same as in ductal cancer. There's no special consideration regarding its treatment, and whatever information is obtained in the next few years from ongoing clinical trials will be directly applicable to this variant. Again, these are absolute leaders in the field, but that was the common knowledge at this point in time. And I think sometimes for at least a few investigators, it's still now. But I think so much has happened to really confidently say that this is not the case, and that really ILC is a unique subtype which deserves independent research, but also not only management, but independent management, also treatment. So what is ILC? The major hallmark is loss of equiterrin. So equiterrin, the gene name is CDH1, is sitting on the membrane of cells. So these are two neighboring cells, and it basically helps the cells to glue to each other. It not only sticks together, but here on the surface, it also signals downstream to the cytoskeleton, which we call basically actin fibers. And it does so through proteins called catenins. This molecule, equiterrin, is lost one way or another in almost each ILC, not in all of them, but more than 90-95%, which is again the hallmark of the disease. As a result of this, the tumors grow differently. So here are like representative pictures of ILC and IDC, also called no special type. The cells in lobular cancers, they grow more in sheets, and there is a less cell-cell attachment. You can see that very often the tumor cells are actually surrounded by more of the stroma, what's in between the cancer cells, in contrast to the ductal tumor or NST, where they are surrounded by other cancer cells. Here's a summary of the distinct clinical features of lobular tumors. Like I just showed you, the cells grow discohesive, more in a string. The tumors are more often multifocal and bilateral. They are more difficult to excise with clear margins. As a result of this, there are more increased rates of mastectomy. Very often the tumors are larger, they're harder to detect and to define the extent of the disease. In general, there is evidence to show lower response rates to preoperative chemotherapy. There are problems with imaging, for example, but that's not the only problem. They are less FDG active. And there are many variants. When we talk about ILC, does not equal ILC. There are many histological variants which are recognized by the pathologist. And more we also understand on the molecular side, there are clearly differences in the molecular makeup. There's unique epidemiology. There's a stronger association with age at first full-term birth, and they are exquisitely hormone sensitive. And this is reflected in response and increased risk with HRT, and I'm coming back to this. And the focus of this talk today is really the recurrence, right? I mean, there are, and again, I will show you more data on this, in general, fewer metastases to liver and lung, but more to unique sites such as ovary, peritoneum, and there are more late recurrences. This is clearly very important. This is, although ILC is only, in quotes, 10 to 15% of all breast cancer, just in the US alone, it's 30 to 40,000 new patients a year. So what about major molecular features? Like I said, the majority of the lobular tumors are ER positive, so they express estrogen receptor. Just as a reminder, the estrogen receptor is a molecule in the nucleus of the cells. It binds to estrogen and then it turns on changes in the cell. So the hormone estrogen binds to ER and basically makes the cell survive and grow. Because they are so responsive to estrogen, that's one of the interesting epidemiological features, is that when you look at menopausal hormone use and risk for cancer, for breast cancer development, that risk is higher in lobular compared to ductal cancer. And this is a differentiation here. This is published by Christopher Lee now almost 10 years ago, and you can see that risk is higher in both hormonal therapies, in both hormone replacement therapies, estrogen only or estrogen plus progesterone. We can model this in the lab. So these are the workhorses for us to study ER positive IDC and ILC. They are cell lines grown in dishes, and there are two ductal cancer cell lines, MCS70, 47D, and these are the two most commonly used lobular breast cancer cell lines. And you can see when we run a immunoblot, where we separate the proteins from the cells on a gel based on sites, and then detect them with antibodies, you can see strong expression of estrogen receptor and loss of VKT1. We can also see this in the cell. We can stain the cell. We can see here that the cells from the cell lines, they express estrogen receptor. It's a brown staining. And when we stain for E-cadherin, which would give you a brown staining, we don't see that in the ILC cell lines. Instead, you have a pinkish staining from a catenin, which moves to the cytoplasm in lobular breast cancer cells and cell lines, as well as in tumors. So as a background, so that was basically my background in lobular breast cancer in general. So now what about metastasis? So the first is really, what about recurrence for patients with ILC? The majority of patients will do really well and the tumors will not recur. We are very interested, and this is a few years ago, to look in a big real-world healthcare data set, basically using cancer registries from 3 institutions here in the Great Lakes area. This is UPMC here in Pittsburgh, Case Western Cleveland Clinic in Cleveland, and OSU, Ohio State University. We collected data on from patients with ductal or lobular breast cancer, and then looked at outcome. How did the patients do disease-free survival? So when did the tumor come back, if it came back, and then what's overall survival of the patient? This was overall more than 30,000 patients. And in contrast to other databases, let's see, some of you might have heard SEER database. This is a limited number of institutions, so somewhat the data are relatively clean, so to speak. As we expected, the majority of lobular tumors are ER positive, 96%. When you then look at outcome, so this is disease-free survival, you can, and this is time in years after original diagnosis, and you can see that these two curves somewhat cross at approximately 6 to 7 years. So basically, after a while, you have more recurrences for patients with ILC. This is not the first time this was described. I think it was the first time with this large number on limited centers. And these increased recurrences translate into worse overall survival. So suggesting that the tumor cells, at least some of them, don't respond to the hormonal therapy. The majority of patients are treated with hormonal therapy, given they are mostly ER positive, the tumors are ER positive, and or that there are aspects of what we call dormancy, that some of the tumor cells find some place in the body to hide and to stay there for many, many years until a decade later, sometimes they get reawakened by mechanisms we don't really understand at this point in time, and then go into metastasis. It's almost, I was surprised. It's almost 20 years ago that the first study was published on a larger analysis of metastasis and specifically metastatic sites for patients with ILC. So this was published in 2004, 20 years ago exactly, where the investigators showed in a relatively large database, and this was actually from San Antonio, UT in San Antonio, that there were fewer metastases to the lung, but more to ovary and GI tract. So we here at UPMC and McGee have a very large database for metastatic disease. This was started by Peg Rosenzweig, Adam Bravsky many years ago. The database has data from more than 1,500 women with metastatic breast cancer seen here at UPMC, McGee, and Hillman Cancer Center. They are prospective, fully abstracted medical records. So we got together and we looked specifically at metastatic sites, comparing patients with IDC and ILC. And so this is actually, I should say, this specific is ER positive disease. So there wasn't a big difference in metastasis to the bone and the skin, but you could see that patients with ILC have significantly lower metastasis in the lung and the liver compared to IDC. And there are more unique sites like the GI, soft tissue mets, ovarian mets. I should say that we also collaborated with a number of investigators here at Hillman and UPMC to look specifically at metastasis to the eye. So this is published a few years ago, where we saw that because of the power really of a large healthcare system and access to the online medical records through the metastatic breast cancer database, that we were able to identify patients with metastasis to the eye was significantly enriched in lobular breast cancer, and sometimes, but not always associated with actually brain metastasis. despite these differences in metastatic sites, I should say that that's not always, that knowledge isn't always shared with the patients. So I want to just show you one figure here from a large study, and I'm sure there are people from you online right now who have helped with that, and I want to thank you from the bottom of my heart. This wasn't an easy thing to do, for either helping to design the study or for participating in the survey and answering it. So this is a survey. You can see that clinicians, researchers, and breast cancer patient advocates from more than 66 countries, more than 1,700 people participated in this. And one of the questions was really patients perceive communications about ILC specific features by their physicians. And one of the questions was, were they told at any point in time that there might be unique metastatic sites, such like peritoneum or ovary, to understand that there might be unique pain which you might want to share. And unfortunately, clearly the majority did not have had this communication. This paper is in revision, so hopefully, this will be out soon, and then I think that will be very interesting to read. We are working with a number of colleagues here to specifically describe metastasis to the ovary. This is the cohort which we work with. We have basically 27 metastases to the ovary. They are mostly ER positive. There's only one ER negative sample. The majority are lobular tumors, but I should say that there are some which are mixed and some which are ductal. And we originally thought, wow, maybe there are some ductal tumors which are actually lobular tumors and they are misdiagnosed. And some of them have EKTR mutation and might gain lobular features. And some of them, they do not have EKTR mutation. They might have other mutation which make them look like lobular tumors. But we are still trying to understand this. So briefly, the tumors in the metastasis, they still look like the primary tumors. This is staining. This is just H&E staining of the tumor. This is a lobular ovarian metastasis. And you can see that these cells grow more on a string and don't attach each other. And again, we don't see the brown staining here, but only the pinkish staining, reflecting loss of EKTR. This is a ductal ovarian metastasis. You can see clearly you don't have this stringy growth, but you have them more growing in a duct, in these tubular-like features. And you have the brown staining for EKTR. And then we also had the mixed cases, where they have both. They have lobular features as well as ductal features. And you can see a little bit here that some cells have more pinkish and brownish staining. The age, the mean onset of or diagnosis of primary breast cancer was 45 years. So really pretty young. It's a pretty young cohort of patients who are later on diagnosed with ovarian metastasis. Disease-free survival in this cohort is 77 months. Survival past metastasis 29 months, and overall 119. So what we are currently doing is we are collaborating with Foundation Medicine to understand mutations in these ovarian metastases. Our cohort is small versus the Foundation Medicine has hundreds of samples, and we try to understand whether there are unique features which drive the lobular cancer to the ovary or the GI tract actually. And we also try to understand mechanism of action. Ultimately, that's critical if we really want to understand how we can prevent or treat these unique metastatic actions. One of the areas we focus on are a potential role of mesothelial cell. It's a specific cell type which covers a lot of organs in the body, like the peritoneum, the ovary, and also parts of the brain, which basically is then actually is potentially related to leptomeningeal metastasis. What about clinical trials in patients with ILC and specifically metastatic ILC? Unless I'm mistaken, I think there's only one trial specifically for patients with metastatic ILC. That trial was the Gelato trial, run out of the Netherlands Cancer Institute and was published last year. It's a relatively small trial, 24, 25 patients, and the treatment was an immune checkpoint blockade drug combined with carboplatin in patients with metastatic ILC. The trial actually was early terminated because of sufficient lack of efficacy, and also because the majority of responses were actually seen. So this is basically staying on drug in weeks, yeah. And the majority who stayed on the drug and had partial responses or stable disease were patients with triple-negative breast cancer. I mentioned earlier that the majority are ER positive. There are only very few triple-negative lobular tumors. In this trial, they were clearly enriched and they responded better. I should say, though, that although the trial overall finished early, was terminated early, there are a lot of very exciting and promising signals in this trial. The study it's a really, really fantastic study. There's a lot of correlative science, and the investigators did a really great job to analyze the tissue and show that there are clearly responses. There's inflammation, there is immune cell activation. So it's not like this is a closed book. I think we need to do more and understand it better. There are clearly challenges for clinical trials in the metastatic ILC setting. First, again, we need to understand biology. If we want to have drugs which specifically target unique aspects like metastasis unique sites or survival of these single cells without attaching each other as a result of an altered cytoskeleton, changed microenvironment, we need to understand this to then say, okay, this is a drug which makes sense in a clinical trial. You have a smaller number of patients so that always complicates the trials. And the detection of measurable disease is often a problem in lobular breast cancer. So how can that be tackled? And I just show you like 2 or 3 different examples and research that's ongoing. Dr. Vida Mukta is a physician scientist, a breast surgeon in San Francisco. She has a very strong interest in lobular breast cancer, great expertise. And one of the recent studies she has shown, this is now actually published, that there is decreased clinical trial enrollment for patients with metastatic ILC. And this is the figure here on the bottom right, where you can see patients with stage 1, 2, and 3 ILC either enrolled in a registry or on a clinical trial. And you can see that patients with stage 4 metastatic ILC, there's a significant difference. They are much more included in a cancer registry, but much less in clinical trials. And clearly the requirement for a lot of those clinical trials is measurement of having measurable disease. And the conclusion of her study is that the enrollment criteria should be modified for ILC. So there's a lot of research going on in this. I mentioned previously that imaging is a problem. I am not an imaging expert, so I'm just sharing one slide here from Dr. Gary Olana from a talk he presented at the ILC symposium in Pittsburgh. And basically, to summarize this, is that in general, more traditional imaging approaches, mammography, but also others have, like FES PET, have less sensitivity for lobular breast cancer. And other approaches, for example, a PET imaging, which uses estrogen as a tracer, so it's called FES PET, might be much more sensitive. There's a lot of research going on there, clinical trials going on in this, and it's a very, very active field, and rightly so, because the imaging documented clearly is a problem for patients with RLC. Another approach is really picking up the disease with liquid biopsies. So for those of you who have not heard or don't know what liquid biopsies are, it's basically a biopsy not of the solid tumor, which might grow in the breast or in the metastatic setting in the liver, the lung, the ovary, the brain, but it's taking blood, and from blood isolating DNA, which is shedded from the tumor. We don't have much research specifically on this in RLC. There are a couple of studies, and this is what Dr. Adrian Lee talked about at the RLC symposium. And just to share two studies here with you. One is on circulating tumor cells. So the tumor sheds cells as well as DNA. And interestingly, although relatively small, the study, but you can see that in this study, patients with ILC had actually more CTC in their liquid biopsies compared to patients with IDC. What about ctDNA? There aren't many studies out there. This is a study published now like 1.5 years ago. In that specific study, they did not see a difference in the amount of ctDNA positivity, but they showed that ILC had a higher number of pathogenic mutations compared to IDC and a lower number of copy number changes. So this is an area I think is really, really promising. It's not, I just want to say it's the future, but it's already the present. But we need to do more specifically in the area of ILC. It's especially important with all the imaging problems. The two slides I just showed you were presented at the ILC symposium in 2023. I realized on the bottom of the slide I made a mistake. It's not 24, but 23. The videos of the presentations are all available on the website. So I strongly encourage you, you can go to this website and you can listen to Gary Olana, Adrian Lee, more on imaging, more on molecular changes in metastatic lobular breast cancer. I have asked Dr. Adrian Lee to share a few more slides with me, which I'm going to show here, just because I think they are important and they really point us towards directions we should follow up more studying metastatic RLC. So bear with me here. This is the evolution of the tumor from the primary to the metastatic disease. So the idea is that there's evolution of the disease over time. The normal breasts acquire mutation, and then you have a number of different clones. There is significant heterogeneity. If the tumor would just have one clone and the cells would all be the same, I would almost be confident to say would have cured it for once and ever. But that's not the case. You have all these different developments, different evolutions. These tumors are not the same. Early recurrence is different to late recurrence. And the different cells are the base of this diversity of heterogeneity, and that allows drug-resistant clones to come up. You treat the tumor, cells evolve, come up with another idea how they can basically get around the treatment. So this evolution of the disease. There's actually molecular subtype switching. It's not often, but it can happen. And this is a study which was presented last year, specifically in ILC, that for some patient, which in the primary tumor had hormone HR positive disease, they switched to HER2 positive or switched to triple negative. In that study, they had access to a second biopsy. So M1 is the first metastatic and then the second, and you realize that in the second, you have further evolution of disease, more adaptation, where then tumors which remain hormone receptor positive at the first time of progression, later on they switch to HER2 positive or triple negative disease. So it is tremendously in flux and tremendous heterogeneity of the disease. In that study, they then they looked at somatic mutations comparing primary ILC, and then also how does it change compared to metastatic disease. This is a summary of a number of publications which have been studied. Some of those we have contributed to. So EKTRN and PI3 kinase mutations are frequent, and but there is no big difference between primary and metastatic disease. They are so-called truncal mutations. They're already in the primary tumor, and then they're in the metastatic disease as well. Other mutations, in contrast, like HER2, estrogen receptor, and NF1 are increased in the metastatic setting compared to the primary tumors. We do absolutely need more data comparing primary to metastatic. Some of those studies are relatively small. Just want to have one slide on HER2 mutations. I'm not going into detail here, but it is an area which is actively studied in lobular breast cancer. There is an enrichment for HER2 mutations in lobular tumors, more in the metastatic compared to the primary. Then specific subtypes like the pleomorphic have even higher levels. Yeah. And here is this is a figure from a large collaboration a few years ago, where we basically looked at the number of HER2 mutations comparing ILC and IDC. And all cases, again, as expected, we have like 15% lobular tumors. But when you look at the case which have HER2 mutations, they are significantly enriched. And if you look at those which are oncogenic, which are really driver mutations of the disease, it's more than 50% are in ILC compared to IDC. A couple of groups are working on this. I just put two references here. Obviously you have access to the slides later on. You can look those up. These are not the only ones, but there's a lot of excitement that you not only have potentially more druggable HER2 mutations in the disease, that potentially the mechanism of signaling might be different in lobular compared to ductal tumors, in that these growth factors which sit in the membrane, HER2, EGFR, HER3, FGF receptor, IGF receptor, that they somewhat signal different, that they are more active and therefore potentially represent more of a dependency in these tumors compared to IDC. And the reason for that is loss of EKTRN. There's different folding of these molecules in the membrane. That's an ongoing area of investigation in a number of labs. So how do we intensify? How do we get more data on metastatic lobular breast cancer? We do think, and this was introduced, Mary talked about this in the beginning, that the rapid autopsy, the organ donation program, is a program which can help with that. It is clearly challenging to access metastatic lesion via biopsy. And the access to tissues for many different organs to study organ tropism. So why does one tumor go to the ovary, to peritoneum, to the bowel, as compared to another one which does not go there, which only goes to the lung? Or you might find some cells, but they sit dormant there. We can only do that if we have access to multiple metastatic tissues. Clearly, more tissue, larger amount of tissue makes it easier to use different assays to study this. At the same time, we can take tissue and we can make models. We can grow them in the lab, and we can study drug resistance, tumor evolution, and like I said earlier, also dormancy. Why do they not grow in some of these tissues for some patients? Yeah. There was a session on organ donation programs at last year's at the Metastatic Breast Cancer Research Conference in Utah last year. Representatives from a number of different programs from UNC, MD Anderson, Utah, Leuven, and myself, and a breast cancer advocate, Stephanie Walker, presented, and there was really, really active discussion on how we can together normalize this process more and help each other and collaborate. Just one slide on our program. It's called Hope for Others. I should say we had it actually called, it was a rapid autopsy program. We had patient advocates involved, and the first thing the patient advocates actually said that we should rename this, that the title autopsy is just not great, and they renamed it into Hope for Others, others standing for our tissue helping enhance research and science. We have, here in the PIT program, we have 102 patients consented. We have performed 38 autopsy. And I should say specifically for lobular breast cancer, we have an ongoing collaborations with the Uptider autopsy program in Belgium to study metastasis in patients with ILC. And that's an actively ongoing collaboration. The patient advocates actively involved, headed or overseen, spearheaded by Christine Hodgson, Stephanie Walker, others are involved. And question they have helped us to ask is, how to address patients and family. What's the role of the treating physician or other caregivers in the consenting? Logistical problems, and how to normalize the process. And I should see part of these studies are funded by Komen, and there's at least there's some, it could be more, but there's some funds given by Komen to support the advocates, and this is both one-time financial, but also just to go to meetings and be there and present the program, which I think I'm very, very grateful to Komen for that. So what about, I want to end with modeling the disease and conclusion and outlook. So in the end, part of the reason why lobular cancer wasn't studied more was really that we didn't have enough models for that. And a few of us got together a few years ago to put together, this was actually spearheaded by George Flomos from Switzerland. It was a Herculean effort to collect all the data out there and put it out in this publication, which is a tremendous resource for everybody interested in lobular breast cancer, no matter if it's primary or metastatic disease. Our team, we are working intensely on all of those, on cell lines, on making models with loss of vegetarian, organoids, mouse models, PDXs. But I just want to share one piece of data with you today on the ILC organoids. So the organoids, this is actually done by Dr. Adrian Lee, who oversees the precision medicine initiatives here at Pitt and UPMC. The tissue, no matter if it's from the primary or from metastatic lesion, is brought to the lab. It's chopped up into little tiny clumps of cells. It's incubated in a little dome of gelatinous matrix, and it can grow in there. It grows much slower than cell lines, but it can grow well. And we can look then, for example, these are lobular organoids, these two, BO46, BO53. And you can actually see, if you compare this to an organoid from a ductal tumor, you can see that even without staining anything, they look more like we call it grape-like. They look like a grape. They look like grapes, right? The cells don't quite attach each other so tightly as a result of loss of vegetarian. And we can stain for vegetarian, and we don't see it in these lobular tumors. So the generation of these organoids, again, very grateful for Komen. We also have then funding from BCIF, so the BCIF Legacy Project, to characterize these organoids, characterize the respective tumor, and put this all on a web portal and share this with other investigators to really make this a tool available for everyone. We have now generated more than 20 lobular organoids, and again, just looking at EKTL mutation, they're the same EKTL mutation as we would expect, as you see in the clinical specimens. And I want to end with an example for using the organoid in a patient with metastatic ILC. So here, and this is again through collaboration with our clinician, in this case with our orthopedic surgeon, who provided access from a patient who consented to give her bone metastasis for research. So we basically, there's a patient who originally had ER-positive lobular breast cancer, unfortunately recurred with bone metastasis, and we had access to the primary tumor, but also to the fresh bone metastasis. And the fresh bone metastasis was taken into the lab and organoids were generated. We could sequence the primary tumor and the metastatic disease. Or here on the bottom of the slide, you can just see the disease progression, the treatment of the patient. And then when we had access to 2 different bone, one in the pelvis and one in the tibia, where we then actually grew them as organoids. You can see pictures here how the organoids from the left metastasis and from the right metastasis grew in the lab, a lower and higher magnification. Both of those grew. We sequenced them and identified somatic mutations. As expected, EKTL mutation, but we also identified mutations in PI3 kinase and in BRCA1. And you can then take these organoids and treat the organoids in the lab with drugs specifically targeting these changes in the metastatic disease setting. And in this case, the organoids were responsive to a drug targeting mutant BRCA1, talazoparib, and we could also take the organoid, transplant them into mice, and then treat the mice with a drug targeting PI3 kinase, AKT pathway, alpelisib, and again, we could see shrinking or stop of growth of the tumor. So this is something where you think you could really do this if you have a great setup of interaction between the surgeons, the pathologists, the translational research lab, and grow these organoids, which really, in most cases, I mean, sometimes we have problems growing them, but when they do grow, faithfully represent the disease. So to summarize, there are many shared, but clearly also unique aspects of metastasis in patients with metastatic ILC, characterized by late recurrences and also unique sites of metastatic disease, and where we really don't understand the reason for these for migration and invasion at these unique sites. There's clearly excellent progress over the last decade. It's not sufficient, but progress has been made thanks to increasing number of researchers studying lobular breast cancer and the work of the advocates. What needs to be done more? I'm sure the list is not comprehensive. There's more, but the focus should be on increased study of evolution and change using liquid biopsies. I mentioned these circulating tumor cells, ctDNA. The jury is out whether it's really more there in ILC than IDC, but I think it looks like that, and follow it over time and see if we can not only pick up metastatic disease earlier, but can we pick up changes, evolution of the disease with respect to mutations, but even transcriptional changes. Is there subtype switching, for example? We need to develop more models to represent the unique metastatic pattern and the heterogeneity of disease. I mentioned this at the beginning. I think our understanding of the differences between the different subtypes of lobular breast cancer and how it correlates with different molecular changes is only at the beginning. We need to model that, we need to work together, because now you have a subtype of a subtype, so the numbers start to be smaller, and we need to have different centers working together. Dormancy, so the sleeping of the cells somewhere in the body, might explain late recurrences, and we really have very limited understanding of dormancy. In the old days, people studied this with some mouse models or other means. I do think the rapid autopsy program, we hope for other programs and the like, can help with that, to try to understand where do the tumor cells find a niche to survive for months or years, and what do they look like? And can we either prevent them from ever waking up again, or do they have pathways activated which we can actually specifically kill these dormant cells? People do work on imaging now in ILC, not enough, we need to do more, trying to understand. There's a big area now on radiogenomics, trying to understand the correlation between imaging features as well as tumor features is a growing area. And last but not least, I think to have clinical trial specifically for ILC is really important. Either, I should say there should be both, that we come up with drugs which target unique dependencies in lobular breast cancer as a result of loss of VKTN. And if we don't have unique drugs, unique pathways, if we test drugs which are also tested in ILC, then at least the clinical trial should have sufficient number of patients with ILC that we can make sound conclusion from the trial results and not have like post hoc retrospective analysis with small numbers of patients. Very important slides. I would like to thank the funding. I mentioned Komen already. I have many years of very productive interaction. Actually, Komen funded me already when I was a trainee. Interaction with Komen, many other foundations, the NIH, the patients for donating blood and donating tissue, growing number of patient advocacy group, which I've really enjoyed with working. Obviously the lab, our lab, which I co-run with Dr. Adrian Lee. Fantastic collaborators here and elsewhere. And the 2024 ILC meeting is going to be in Leuven in Belgium. And I hope that some of you will be able to make it there. And on that note, I will stop here, and I hope I am back. I stopped sharing my screen, is that correct?
Yes, thank you. That was amazing. I'm thanking you for everybody. Lots of positive comments on how clear that was to understand. We have received a few questions. I'm going to start off with a question from me. You talked about the heterogeneity. Has anyone looked at that in the primary tumors and the lymph nodes? And I guess related to that is, given that it's typically slow growing, is the mutation rate higher or lower than other types?
Yeah, Mary, great question. So there are a few investigators who clearly have looked at the heterogeneity. And I mean, for example, there's a group at Memorial, George Rice Fellow, and many others who try to understand the molecular features of these different histological variants. That has not been looked at with their respective matching lymph node. So basically your question is, does the subtype remain the same or not? So very small studies. I can tell you that our group, we have looked at mixed ductal lobular cancer. A similar study was done recently by Christine Desmet in Belgium, also looking, when you have a mixed primary, is the responding metastatic disease still mixed, or is it lobular? And very often, actually, has more features of lobular disease in the advanced setting. But I do think this is an area we need more focus on. We need to sequence those samples to see whether there are unique changes in these different subtypes. The pleomorphic is an example where I think we have learned from the HER2 mutations are specifically enriched in the pleomorphic setting.
One of the questions we received was, what are pathogenic mutations in ILC and are they actionable?
So there are clearly pathogenic mutations which make the tumor more bad, so to speak, right? They are pathogenic, they make the tumor bad. And examples for druggable pathogenic mutations are PI3 kinase, it's a prime example. I think another example is HER2 mutations. Other examples are, just like in IDC, we have frequent ER mutations in the metastatic setting. I should say there was actually a study published to look specifically at whether there are differences in ER mutations between IDC and RLC. That does not seem to be the case. The AKT, the PI3 kinase AKT pathway seems to be specifically activated, more activated in lobular cancer than in ductal cancer. So we have more mutations also in AKT and loss of PTEN, other pathogenic mutations in lobular breast cancer, for example.
Okay. You talked about evolution of disease. What do we know about the evolution of disease for de novo diagnosed metastatic ILC patients?
Yeah, that's a fantastic question. That's a really good question. So, yeah, that's a great question. I don't know the answer to this. So, I guess the question is either the biology or the outcome different for de novo ILC, the novo metastatic stage 4 versus acquired? And I think that we don't have the answer for this because we just don't have the numbers. We really don't have sufficient numbers. I don't think, we haven't sequenced that large number of metastatic ILCs. There are a couple of studies out there. I think it would be really worse. I don't know who asked the question, but I think that's a fantastic question. I wonder if they could go back and actually look at that if the numbers are. Yeah, it's a great question.
I know we're running out of time. This one might be fast, or we might have to move on to closing. But you mentioned HRT. Has anyone looked, is there an association with birth control use earlier?
I don't know. I would say whoever's interested in this, there's a scientist at the Hutch, Chris Lee is his name. He has really looked a lot into epidemiological aspects of lobular breast cancer. I think if anybody would know the answer in a detailed, informed way, he would be the person to ask. And actually, while I'm speaking, I should say that part of the prior question, or two questions previously, was also just frequency of mutations, right? And I should say that we worked with Foundation Medicine a few years ago. They're very interested in collaborating with academic folks to study different aspects of the disease. And the numbers they have from liquid biopsies, from sequencing, are large. And we identified that the overall mutation rate in metastatic ILC is actually higher than IDC. There seems to be, at least for a subset of metastatic ILC, a really high rate, potentially through a mechanism we call ApoBac activated, activation of mutations. Again, it's an area we don't understand enough, but there's clearly a subset with high rates of mutations. which then makes you think, wow, could that also be a subset where potentially you have still opportunities for, role of the immune system, yeah.
Well, thank you. That was wonderful. I'm going to turn it back over at this time to Kelsey.
Thank you so much, Mary, and thank you for a fantastic presentation, Dr. Osterreich. I want to take a moment to thank everyone for joining us today for this really important event on such an important subject. We want to just express our biggest thanks to these wonderful speakers. Again, that's Dr. Steffi Osterreich and our moderator, Mary Domenecki. I want to thank everyone for these really amazing questions that have been answered and submitted. And if we didn't have a chance to get to your questions on metastatic ILC, please submit them in the Q&A and we will work with Dr. Osterreich after the fact to get your answers.
Yeah, and I would be happy to work with all of you with Komen, and if any questions, please don't hesitate to reach out.
We really appreciate it. There was a lot of great questions in the chat and we didn't have time to get to everything. But the good news is today's session was recorded and will be posted on our landing page as well as our YouTube channel soon. So you can rewatch this session or catch previous sessions that you may have missed. Answers to your questions will be mailed out through to the list of everyone who registered, along with a link to the recording so you can catch it later. Please, if you enjoyed today's session, please help us spread the word about this important content to your friends, family, and healthcare professionals. Once more, we'd like to acknowledge the sponsors of the 2024 MBC Impact Series. Their generous support allows us to bring this community together to share this critical information, important resources, and personal experiences. Thank you once more to our premier sponsors, GE Healthcare, AstraZeneca, Merck, and Gilead, and to our signature sponsors, Walgreens and Daiichi Sankyo. This series is made possible because of their generosity. Please also help us by completing a survey. You can find a link for this in the Zoom chat. Your feedback is critical and helps us to plan future MBC programs. Finally, the next event in the MBC Impact Series will be on June 20th, and it will be focused on metastatic HER2-positive breast cancer. We look forward to having you log in and join us with the conversation. Thank you so much for spending this time with us today, and thank you to everyone who made this event possible. Stay safe and stay connected.
Bye-bye.